Each stereocilium has a stretchy protein fiber called a tip link, which is attached to its lateral side and then to an ion channel of an adjacent stereocilium. When the stereocilia of auditory hair cells bend, the tip link tugs on the mechanical gate of the ion channel, causing it to open. Since the cochlear duct is filled with endolymph, which is similar to intracellular fluid, potassium ions are high in concentration outside the auditory receptor cell. So it flows down its concentration gradient, depolarizing the membrane of this receptor. The receptor then releases neurotransmitter to the dendrites of the cochlear nerve. If a strong enough EPSP results, an action potential is produced and a nerve signal will travel along the cochlear nerve, which is a branch of cranial nerve 8. It also serves as the first order neuron of hearing. Each ear sends fibers to both sides of the medulla oblongata in locations known as the cochlear nuclei. Here, the cochlear neurons synapse with the second order neurons that ascend to the superior olivary nucleus of the pons. The superior olivary nucleus functions in binaural hearing, which means comparing signals from the right and left ears to identify the direction from which a sound is coming. Other fibers from the cochlear nuclei ascend to the inferior colliculi of the midbrain. The inferior colliculi help to locate the origin of a sound in space, process fluctuations in pitch, and mediate the reflexive movements of the head and neck in response to loud noises. Third-order neurons begin in the inferior colliculi and lead to the thalamus. They synapse in the thalamus with fourth-order neurons, which travel to the primary auditory cortex in the superior margin of the temporal lobe deep within the lateral sulcus. The temporal lobe is the site of conscious perception of sound, and it completes the information processing essential to binaural hearing.